Okay, so the topics I'm going to be covering, excuse me. Please let me know if I'm not loud enough. So I'm working on a project that we're just in our first of 3 years on, and the goals are really to understand, kind of get a first level of understanding for resistance to parasites in sea louse. So we're probably a number of steps behind in terms of comparing this to some of the other systems you're working on. So I think that we have a lot to learn. I have a lot to learn from you all about how to proceed in really understanding the evolution of resistance in this system. My role in this project is kind of to go from, I'm the modeler on the project, and so one of the challenges I'm finding is to translate some of the data that we're collecting. A lot of it is really qualitative genomic information, and how can we take that data and put it into a quantitative model. So I'm hoping that you all can kind of keep that question in mind as I present just the general approach that we're taking over 3 years, and I'll share the data we have available, but this is kind of in process right now. So the system that we're working on is sea louse. They're parasites of salmon. They're crustaceous copepods right here. So these sea lice feed on the mucus of the salmon, and occasionally, as you see in these gravid females, they actually feed on the blood of the salmon. So this can lead to secondary infections, and with high enough levels of infestations, this can cause mortality in salmon. So salmon are farmed in these open net aquaculture systems, and because they're open to these coastal waters, there's a lot of opportunity for exchange of pathogens and parasites to other farms and also to wild salmon. We have a lot of the areas that are best for growing farm salmon tend to be areas where we have a lot of wild salmon. So we have these kind of ephemeral refugia when the salmon are migrating through these systems. The life cycle of the sea louse is fairly complex, but it is divided into two main strategies. As young nauplii and copepodids, these are free-swimming planktonic organisms. So that's when they're moving throughout the systems and have the potential to infect wild and other farm salmon. After that, they find a host and attach, as we see here. So costs of these infestations on salmon farms are estimated at $500 million to the industry every year, so pretty substantial. And another major concern is that infestations have been associated with declines of wild salmon. So a number of methods are used to control sea lice infestations. We've got a lot of integrated pest management, so we have the identification of these bay management areas. So they're usually bounded by hydrological boundaries, and within these areas, we coordinate the movement of salmon into the aquaculture pens so that we have single-age cohorts going through, and following that, we will fallow the pens so that we can decrease the abundance of sea lice in these areas. We also use cleaner fish, or wrasse, that can eat the sea lice off of the salmon hosts. So we've got a number of different approaches for controlling sea lice, but we're very heavily reliant on chemotherapeutants. So a number of chemicals are used to control sea lice, and resistance to every chemical that has been used has been found in at least one population. So we see a lot of evolution of resistance, and in many cases this is quite rapid, occurring in less than 8 years after these chemicals are in use. So some of the chemicals we're seeing, for example, emamectin benzoate, this is one that I'll be talking about quite a bit. They vary in their mode of delivery. For this one is an in-feed, so it's given in the food to the salmon. So it can take quite a while before we see any effects on the lice, in this case 7 to 60 days. We also see a lot of differences in the life stages that are targeted. Some of these chemicals are only lethal to certain stages. For example, these chitin synthase inhibitors really only affect sea lice before their final molt. So understanding how this variation in the mode of action, in the mode of delivery, and the life stages targeted is one step we can make towards understanding why we get different rates of resistance evolving. So to really understand this process, we need to know information at several different levels. For example, at the molecular level, what is the mechanism of resistance? Is it genetic? Is it epigenetic? How many genes are involved? At the individual level, we need to understand the life history and behavior of this organism. This is just results from a population matrix model we did showing the life history of the sea louse. And what you see is that we have different temperatures here, and this is our population growth rate. So as we increase the temperature, the population growth goes up dramatically. And this is due to increases in reproduction as well as decreases in generation time. So in 4 degrees, we have over 100 days of generation time, and it's about 25 at 20 degrees. So a 4-fold decrease. So that's really important to take into account when we think about the life history and the evolution of resistance. At the level of the population, we don't know very much about the immigration and emigration of sea lice into these different populations. What are these rates? Are the individuals that are coming in, are they coming from wild populations or farmed populations? In addition to infesting salmon, these lice also infest stickleback, and these have very different life histories, very different spatial temporal patterns than the salmon, and we don't know anything about how that affects movement of sea lice. Finally, we want to know at the environmental level how these factors like temperature, salinity, and these exposures to treatments is influencing our populations. And so this is a lot of information, and I think this is really where modeling comes into play, because we have complex interactions occurring on different time scales. They're nonlinear, and we can really explore a lot of different scenarios when we integrate these levels of information into our models. So in our system, our major knowledge gaps are at the molecular level in terms of what are the mechanisms of resistance. And then at the population level, we know nothing about the population genetic structure. So the goals of our project are first to fill these gaps. We want to characterize the population genetic structure. Secondly, we want to identify mechanisms of resistance, or at least have a few targets to explore further. And then finally, we want to incorporate this information into a model that can explore variation at all of these levels and that is flexible. So as we gain new information, we don't have to build a completely new model to incorporate that information. So for our first goal, I'm just going to rapidly sort of go through generally what our approaches are. It's not a very in-depth talk. It's more of just a broad overview of our approach. To look at population genetic structure, this is Ben Koop. He's a professor at the University of Victoria. He's really targeting this question. And this has actually been addressed in the past using microsatellites, and there was no evidence for any population structure in either Atlantic or Pacific populations of sea lice. And we have no idea if this is a real signal or if this is just due to the loci that were used and just the resolution of the study. So Ben is taking this up a number of levels and using a genomic SNP approach to quantify population genetic structure. He and collaborators in Norway have sequenced over 40 sea lice, and they've identified more than 800,000 SNPs. From these SNPs, they're creating a 200,000 SNP chip, and they're going to use this to characterize population genetic structure. So what can we learn? We can see if there are mutations associated with drug resistance. We can compare results from SNP chips in field and lab collected lice and hopefully resolve some of those issues about whether signals we see in the lab are also occurring in the field. We can look at the heritability of resistance using breeding experiments. And then we can address the population structure and try to estimate immigration, emigration rates, and mixing between farmed and wild hosts. So that's it for that section. We're really in the process of just building this chip now, and we have no data at this point. Secondly, we want to identify mechanisms of resistance. We're using a number of different approaches here, but most of them are based on sea lice in eastern Canada. So we've got the New Brunswick, Maine border here. This is the Bay of Fundy. These are bay management areas where we have farms in black, and some of these areas have had farms for quite a while. Other areas, such as these Grand Manan sites, are quite new. And so we see very little resistance here, or it's just starting to show signs of resistance, whereas resistance to emamectin benzoate is quite strong here. So we can use these populations to examine the mechanisms of resistance. This work is being done mostly by Mark Fast, who's a professor at the University of Prince Edward Island. So he did an EC50 with populations. I'm just going to show the results of two populations. So this is a resistant population from Bay Management Area 2A and a susceptible from Bay Management Area 2B. And you can see that the EC50 is much higher in the resistant population, but we also have a very large difference in susceptibility between males and females. Also, quite large confidence intervals, even in the susceptible population, suggesting perhaps there's already the variation to select upon. Oh, excuse me. So what we did next, or what Mark did next, was to look at gene expression in these populations. So he could identify potential mechanisms of resistance. So doing a transcriptomic study, this is a principal components analysis showing the separation of gene expression in our susceptible and resistant male and female populations. There's between resistant in blue and susceptible males and females in red. There's a lot of separation, as well as between males in the squares and females in the red, or circles. So if we want to look at more specifically what those genes are that have differential regulation, in our resistant populations, we see very few shared genes between males and females, only 6 of more than 200 genes. And similarly, in our sensitive populations, there's only 10 shared genes here. When we look at what these are genes that pop up often when we look at stressed organisms, and so we think they're associated with general stress responses. So that gives us at least some clues as to what genes we can begin to look at for mechanisms of resistance. Finally, we're also interested in epigenetic changes in these sea lice. So there's a lot of evidence that pesticides can alter methylation patterns in the genome, and that this can alter gene expression. There's evidence showing that in some cases this can be adaptive, other cases there are very high fitness costs. So we really don't know whether methylation is occurring, and if it is, what is the net result on fitness. So right now, what we're doing, I'll explain this first, actually. So what we see, just kind of as a schematic, very simplified, we have these upstream CPG islands where our methyl groups can attach. And if you have methylation occurring, that can alter, in many cases, suppress gene expression. So these changes can cause rapid responses to environmental changes, such as Jess had discussed, and they can be heritable. So to look at whether we're seeing changes in methylation, we're just doing a methylation-sensitive AFLP analysis, which will let us see if there's just genome-wide changes in methylation without knowing in any detail what those genes are. If we see these patterns, then we can follow up to look at whether what genes are associated and if there are fitness costs to the genes, or perhaps they're adaptive. So that's our data collecting stage of the process. We obviously have a lot more data to get, and I think one of the challenges is that there's going to be an overwhelming amount of data, and how do we take a lot of this data and really pull out what's important and put it into a comprehensive model that can let us explore questions about how do you manage for resistance. So the model that we want to build is, or we're working on, is we really want it to be very flexible. And when we look at different modeling approaches, we need to be able to incorporate a lot of factors on a lot of different levels. We want to look at stochasticity. We want to look at behavior and individual variation. We want selection events that can be instantaneous or constant, such as the cleaner fish. And really, individual-based models are the only approach that is flexible to incorporate all of this information. So that's what we used. Individual-based models kind of operate on two levels. So you can characterize what's happening within an individual, and that's really what you model. And then you can look at population-level characteristics, such as the proportion of resistance genes in a population, or population changes in population size over time. So the model that we built is using the AnyLogic software, which I highly recommend. Even if you're not a math person, it's very intuitive. And it's based on a model we previously published about controlling sea lice with cleaner fish. And this model is actually online, a simplified version of it, and you can actually change parameters and see how that affects the population dynamics. This is kind of a nice tool for managers because they can actually kind of test different scenarios for control and see how that affects, what kinds of effects it has. So our model has two levels. Like I said, this is the individual level. Right here we have our state charts for the sea lice. So our sea lice can go through different life histories. So we have the egg, the nauplii, the copepodid, then they attach, and we separate into males and females, and we have breeding down here. So at every transition, there's a probability of death, and then there's a probability of developing to the next stage. And these developmental rates are all temperature dependent. As I showed you before, temperature is very important in controlling the generation time, so we needed to include this in the model. And then with each individual, we can assign them genes with certain levels of resistance, and that can change the probability of death. For example, if we have a treatment that attacks these mobile lice, if they have the resistance gene, that will change the probability of dying. At the upper level of the model, we can control variables that affect the entire population. So at the environmental level, we can have different temperature scenarios, cool temperatures, warm temperatures. In terms of our treatments, we can have different values for our treatment efficacy. We can have different algorithms for when treatments are applied, depending on the level of number of individuals in the population. We can have in-feed treatments. We can have cleaner fish. And then we can also alter our population dynamics. We can change the number of external eggs coming in, the proportion of those eggs that are resistant, and then the probability that our larvae will actually find a host to attach to. So this is very highly flexible, and as we get more data about population structure, for example, we can alter some of these parameters. In terms of the genetics, we've just set it up at this point. It is a one gene, two allele system, so we can have parameters for the benefit of our resistance alleles and if there's a fitness cost. Then we can collect data on the number of susceptible, heterozygous, and resistant individuals in the population. So from preliminary results, these are just from single runs. Because this is a stochastic model, you really need to run it a number of times and estimate confidence intervals. But just to give you an example of results, we ran our model in a cold year and a warm year. Over time, this is over time, we have the number of lice per host, and it's divided by, it's shown for each different stage, which isn't so important to know, but what you can see is that, hopefully you can see these steep drops. We have peaks and then they drop, and that's where treatments are applied. So in our cold year, we had 7 treatments applied, and our warm year, we had 13 treatments applied. And this is just based on the lice reaching a certain level, and that tells the model, apply a treatment. If we look at the genetics of our populations, we have heterozygous in orange, we have susceptible in black, and then resistant individuals are in red. So we can see the proportion of resistant or heterozygous individuals in the population. In our cold year, we have some increases in resistance when those treatments are applied, but by the end of the year, the level of resistance is quite low. In comparison, in our warm year, we have a much higher increase in the number of both heterozygous and homozygous individuals. And so this is kind of confirmation that our model is working as we expected, both because we have greater selection with more treatments and also because we have a much faster generation time in the warm years. You would expect resistance to evolve faster. So this is as far as we've come in the project. Hopefully we'll update you as we get more data. But to conclude, we're finding a lot of variation in sensitivity in our populations. And this is both across gender and across populations. We have evidence that this can be inherited. And we have these individual-based models, which seem to me to be a very flexible platform for studying evolutionary processes and incorporating processes that are at different levels of organization. So future directions are to continue to develop our SNP chip and hopefully to estimate some of the population genetic structure, investigate potential mechanisms of resistance. These can be mechanisms that we find based on our SNP chip or based on gene expression from our methylation studies or our transcriptomic results. And then in terms of model development, in the absence of the data to really parameterize the model well, I think there's still a lot of questions we can explore. So we can look at how does the immigration and emigration rates affect the evolution of resistance, depending on what stage or sex we target with the treatments, how does that affect how fast resistance evolves. And then we can also look at effects of temperature. And so here's our team. It's a really large group, and we have funding from Innovation PEI and CERC and the Atlantic Veterinary College. Thank you.